Unfortunately, David is not feeling well, so we're missing his talk. Jay didn't offer to take his spot. Okay, so we have Abhinav Checkervarty. Abhinav, I didn't tell you you're on next, but you're on next. All right. This is supervised by Dr. Scott Tebbett, who's a long-term colleague and friend over at the Heart Lung Institute, just down the street kind of thing, isn't it? Okay, enough.
I'm going to present my research titled Sickle Cell Trait Affects Early Life Immunity but Not Vaccine Response. So sickle cell trait is a genetic condition which affects over 300 million individuals worldwide, with most cases originating from Africa. This condition is considered benign, but complications can occur under certain environmental stressors, such as lack of oxygen or extreme exhaustion. So in this study, we tried to explore the effects of sickle cell trait on early life immunity. And to do this, we use data from a vaccine study. This study was with 720 neonates who were born in the Gambia and vaccinated with hepatitis B. So 2 blood samples were collected from each neonate, one at the day of their birth and one on a randomized day, which was during the first week of their life. These samples were then used to generate multiple omics data sets, or the biological data sets. And in addition, we used 2 additional samples to record their anti-hepatitis B antibody titers, which was recorded for after 1 month and after 4 months. So we hypothesized that we can use the reads, which are mapped to globin genes and usually filtered out in blood transcriptomic studies. We can use these reads to identify neonates that have sickle cell trait. And the neonates with sickle cell trait exhibit differences in their immune system during early life and their vaccine response. So firstly, to identify neonates with sickle cell trait, we genotyped their reads, which were mapped to the globin genes. And we used both birth and non-birth samples to make sure that our genotyping pipeline is giving consistent results. And we identified 118 neonates had sickle cell trait, which is around 17% of the total. And then 4 neonates were homozygous for sickle cell disease. Now let's start by looking at their vaccine response. Here I'm showing a plot of their anti-Hep B antibody titers after 1 month and after 4 months on the top row. And on the bottom row, these are the same titers, but adjusted for maternal antibodies. So we found that the vaccine response is not different between the wild type and the sickle cell trait group. And then to assess the impact of sickle cell trait on early life immunity, we used biological data sets or omics data sets and analyzed them using supervised machine learning models. These models were designed to predict neonates if they have sickle cell trait or not. And then we investigated the omic features which they selected as predictive of sickle cell trait and analyzed the biological mechanisms which were different. So we used 2 methods, which was single omics and multi-omics integrated data. In multi-omics, we can also assess the correlation between different biologies. So we found that the non-birth sample models worked better in prediction, and the birth samples were less predictive. Especially on day of life 3, the prediction reached the highest performance, and the average area under the curve was 69%. So using findings from this model and the single and multi-omics model and other analysis, we looked at the pathways which were enriched in sickle cell trait. So we found that the pathways associated with blood and platelet function, transcription factor which relate to oxidative stress or some hematological malignancies, and pathways which were associated to liver or kidney were differentially expressed in sickle cell trait. And finally, we also were able to get a validation data set. This data set was a smaller data set, but independent cohort from the Gambia for a different study. And in this validation cohort, we identified 7 neonates which were sickle cell trait, which comes around to be 16%, which is very close to our original finding from our original cohort. And the models which were trained on the original cohort predicted the neonates that had sickle cell trait in the validation with very high accuracy. So in summary, we used the usually filtered out blood transcriptomic data to identify neonates with sickle cell trait. And in this data, the neonates with sickle cell trait demonstrated similar vaccine response to the wild type. And we also identified some pathways which were altered that relate to blood and platelet function, transcription factor, which are associated to hematological malignancies, oxidative stress, and liver and kidney. Thanks.
It's on. That was great. I just had a question. Would you have thought that patients with sickle cell trait would have had an altered immune response in terms of vaccine response? Because I'm not a clinical hematologist, but all the sickle patients probably receive immunization early as kids, don't they? Maybe I'm asking that too differently. Go ahead.
In the literature, we see that there are clinical reviews that tell that sickle cell trait do show some immune-based differences. Included with in JAMA, there was like COVID-19 vaccines work differently in people with sickle cell trait. So there was like literature available that they have different immune system. So it was worthwhile to see if they also have different vaccine response in hepatitis, and does it come in early life?
Thanks very much.